Travis Schaeffer. Travis has a long history here at South Dakota State University, doing his master's and his bachelor's here in this department. And he's currently an employee with USGS working out of Yankton. And today Travis will be telling us about modeling fate of the soybean isoflavone genistein in aquaculture settings, a fugacity approach.
Dr. Leffler. It's fun saying those words about 3 times fast every day. So, as you mentioned, my name is Travis Schaeffer, and I'll be taking you through some of the proposed research I'd like to perform during my dissertation. So just a little bit of background. Aquaculture, or the growing of fish for consumption purposes, has been seeing an increasing trend over the past several decades. And a part of this trend has occurred due to the fact that a lot of these fish are being fed aquaculture-based diets in order to increase their growth. And these aquaculture diets, it becomes very essential to determine the dietary ingredients used in there, with a big emphasis on protein. And a lot of research has started to grow towards more plant-based proteins due to the fact that they are readily available and they have a much more decreased cost compared to a normal fish meal-based diet. So soybean meal has gained a lot of traction due to the fact that, similar to fish meal, it can have a high protein content and also a very well-balanced amino acid profile that can be utilized by these fish. However, unlike fish meal and similar to other types of plants, it may contain anti-nutritional factors that decrease the overall performance when it is being fed to fish. And one of these factors is called phytoestrogens. So a little background on phytoestrogens. These are naturally produced estrogenic compounds that are found in plants that can induce various types of estrogenic responses in different vertebrates. And there are currently over 100 molecules, and they can be classified down based on their chemical structure. And the main class we want to look at is isoflavones, because these are found primarily in legumes. And during the processing of taking a raw soybean meal and placing it and making it into a soybean meal, most of those isoflavones are found to have been transferred over into the protein fraction of that soybean meal. So within isoflavones, there are several different types of derivatives that can be derived, or that occur. There are several types of glucosides, but of primary importance are these unconjugated forms called aglycones. And the reason these are important is because they maintain a much more similar chemical structure to that of estradiol. So when you're seeing physiological effects that occur from these type of chemicals, it is primarily due to these derivatives within the isoflavones. There are 3 types of aglycones that you can find. There is genistein, glycine, and diazine. And genistein tends to be the most prevalent when looking at soybean meals. So as I mentioned, due to their similar structure, they have produced estrogenic effects similar to that of estradiol. And some of these have included the being able to induce vitellogenesis within fish. Along with that, they've also seen that it can create skewed sex ratios when it is fed to a captive population. In vitro studies have shown that activated sperm in the presence of genistein can actually ends up reducing the overall quality of mortality, leading to reduction in fertilization rates of fish. And probably the most interesting and poignant is the fact that if it is included in a diet at high enough levels or in the water at high enough levels, you can see results of intersex characteristics occurring, where these immature oocytes are actually forming inside of testes of male fish. So some of the potential issues we got to face here is that we have these isoflavones, which are in the soybean meal, and if enough make it through the processing of these aquaculture diets and are fed to fish species, there can be potential physiological effects that occur. But one area that maybe has been looked at or undertaken is the fact that not 100% of these aglycones are probably being uptaken by the fish or bound within that system. So the question is, any unused isoflavones, is there potential that they could maybe get put out into the environment to cause some potential environmental contamination? And will they be at levels high enough to actually affect and cause some type of effects upon wild populations of fish? So one way to kind of get at this, or at least to kind of maybe estimate the amount of genistein that could potentially be within the system or get out of the system, is to use a concept called fugacity. Fugacity basically uses phase equilibrium thermodynamics, where a chemical that is introduced into a system will try to seek and find equilibrium amongst all the various phases within that system. So the overall quick goal and use of using fugacity models is you start with a known amount of concentration being introduced into your system. From that, you try to determine the overall fugacity, which basically depicts how does this chemical, how will it move throughout the entire system. And from that, you use background fugacity criteria for the way that chemical will partition into these various types of phases. And what I mean by that is certain chemicals that are very insoluble in water will try to seek out either moving into air or into the biota to find the lipids and the fats to adhere there. So by knowing these by knowing this background information, you can then use those to calculate estimations of what those concentrations may be as they will separate out within that environment into the other phases. And the reason this is such a useful method to use the fugacity model is that for organic chemicals at low levels, which is what you find most of these estrogenic compounds to be, there is a linear or near linear relationship occurring between concentration and fugacity. And it's depicted by the formula concentration equals fugacity times Z. So Z becomes a very interesting point to take into consideration because it is known as the fugacity capacity. Kind of a fun little term to say all the time. I see Snickers right in the room. And but basically, fugacity capacity is a phase's ability to absorb a chemical. So a phase that has a high fugacity capacity will be able to absorb a lot more chemical versus a low Z value one. So it basically helps you determine how it partitions out amongst the environment. And this can be based on partitioning coefficients. So if, for just an example, if you place an amount of a chemical within this two-phase system, you allow that system to come to equilibrium. You can then determine the concentrations in phase 1 and phase 2, and by dividing those, you come up with this partitioning coefficient. Then by substituting in your ZF values for the C's, you see that the overall fugacity of the system is equal, so the F's drop out, leaving you with Z1 over Z2. And this becomes a very handy way of determining the various Z values throughout your system, because they can either be estimated by knowing the chemical's background, its solubilities, or it can also be determined empirically. So some of the reasons I would like to undertake this research is the fact that, as I mentioned, we're seeing increased aquaculture occurring, and with the use of more of these diets, so there's going to be a high amount of isolavones potentially being introduced to these culture fish species, or even potentially introduced into environments if it is able to get out via effluent waters or through waste products. Along with that, several studies have looked at how soybean meal processing to create human products such as tofu and temp it does have effects upon the isolavone content, or the amount of the profiles, how they can switch from glucosides to algae cones or back. However, nothing has been looked at within the aquaculture realm as what occurs with these isolavones when they are being processed into diets. Along with that, we see that there's the physiological effects that are occurring from both dietary and waterborne. So there is while some knowledge is out there, there remains a vast vacuum of what these can actually potentially do. Various studies have looked at numerous different species, or have looked at different species at different age groups, but there's still a lot of information that needs to be gained to get a better handle on how these chemicals affect the fish. And as I mentioned earlier, not much has been looked at as far as potential contamination issues due to effluents and waste products getting out of systems. So I have 4 objectives to the study I'd like to undertake. The first would basically be to determine what is occurring to the isolavones as they are being processed from a soybean meal all the way through an aquaculture diet. Second, we'll then look at and determine any partitioning coefficients that will be necessary for our models. And that is mainly going to be looking at how is the genistein that is bound up in the feed, what is the interactions that has with when it is exposed to a water source. Along with that, we'll then be taking and developing that fugacity model in order to basically quantify and determine where is that genistein we want to factor out within this system that we're going to propose. And then also determine is this relevant, is it an accurate model. And then finally, we'll also determine what potential physiological and growth impacts will feeding genistein to these cultured fish cause on them. And we'll use a reference diet to kind of compare these results. So just real quickly, the complementary null hypotheses for this research will be that we'll see no differences from from diet production amongst the isolavones. We'll see no transfer of genistein from the diets to the water. There'll be no statistical difference between our model results and our empirical data, and that there'll be no differences in performance or physiological or morphology from culture fish fed these diets versus the reference diet. So diving into the methods I would like to undertake for this, there are basically 4 different phases of this research I want to undertake. In phase 1, we'll look at using we'll basically try to determine what is occurring to the isoflavones and primarily genistein when it's being processed. And in order to do this, I'll be utilizing 2 mass balance studies. And the reason there'll be 2 of these is because current processing techniques, the processing techniques for soybean meal into diets has continued to evolve, and now there are actually preconditioning treatments that can be done in order to enhance the overall protein. So you can start with your regular soybean meal and by adding several different ingredients and also several different steps, you can come out with an enhanced soy protein. And this protein is more readily available and better utilized by the fish when it is included within a diet. So the objective now is to basically pull duplicate samples from each of these points, from the original ingredients and also between each step, to determine what is occurring to the isoflavones. Are they being switched between the derivatives from glucosides to alkycones? Or are they being lost? Are we losing them out into our secondary type of products? Or are they being maintained, and at what levels are they being maintained in our enhanced soy protein? All these samples will then be freeze-dried in order to keep them on a dry matter basis, and we need to freeze-dry them in order to keep the chemical compositions the same, so nothing occurs to the chemical structure from heating for drying. And then from that, we will determine the total isoflavone levels and the profiles, the different type of derivatives that are in there, and also with a primary interest on where are the genistein levels and what's happening there. The second experiment will be basically identical to that, except we'll take our enhanced soy protein and we will add our several ingredients of vitamins and lipids, and we will then process them all the way to an aquaculture feed. So sample points will be the same, and we will do the exact same type of analyses on these as well. So just some background statistics. We'll basically calculate a percent loss between each step in order to kind of get a big, broad view of what's happening here, what steps are causing the most loss or the most change within these. And then in order to determine if there's any differences occurring, we'll place these through a one-way analysis of variance. And if differences are found between these steps, we'll use a least significant difference test to determine where that's occurring. So for phase 2, basically to look at what happens when what happens to the genistein found in the diets when it is included within the water. So there'll be 3 experimental diets we'll be looking at. The first one will be a standard rainbow trout aquaculture diet that we will produce within phase 1. And we'll call this our base diet. And from that, we'll use increasing levels of genistein included at 2 and 4 milligrams of genistein per gram of feed. We'll perform duplicate samples, and basically this will be a static test where we include known amounts of water and known amounts of feed, so we know how much chemical is being added to the system. And we will also hold blank samples holding no food or chemical in it, and also spike samples for some quality control of our analytical procedures. We'll collect water and feed samples from each of these samples, and we're going to do this over a time frame of 15 minutes. 15-minute intervals up to 90 minutes. And then each of the water and feed samples will be analyzed for total genistein levels. And the reason we'll take this data, and then we'll end up calculating that partitioning coefficient between the pellets and the water, using that same type of step as we did before, where we use the concentration divided by the water to come up with that partitioning coefficient, which is a necessary item that we'll need in order to propose and in order to build our fugacity model. And then the reason we are doing a time series of 15-minute intervals is we want to examine the overall relationship that possibly occurs between these feed and water. Basically, is there a rate of transfer occurring of the genistein from the pellets to the water? Are we seeing it that it remains completely bound in the pellet, or is it being leached into water, and at what rate? Because that can determine the pathway that it becomes exposed to fish or potentially gets flushed out of the system. So phase 3 then will be to develop our fugacity model. And basically this model will just help us determine how will genistein, when it's introduced into our system, how will it partition out? What phases will it like to go to? And we can estimate at what levels will it go to. So in order to do this, we have a 3-step process we're going to use. The first would be just to determine what compartments are we finding within this, within our system. So fairly straightforward. We have 4 compartments. There will be the fish in the system. There'll be feed and pellets. There'll be the water, and there'll also be the air. Then for calculations, you also need to know the volumes of each of these. And by utilizing an aquaculture system is very handy for this type of research, due to the fact that we know all the values. They're all recorded. We know how many fish we're stocking. We know what sizes they are. We know the feeding rate that's being included. We know the water levels, and we also know the interactions that are occurring from the interaction surface created on the air and water, the size of that, where there could be some potential interactions occurring with that genistein. So step 2 then is going back to those wonderful fugacity capacities and determining what are those for each of these compartments. So again, we have our same setup, and as I mentioned earlier, these Z values, they can either be estimated based on the chemicals' solubilities or what you can find in the literature, or they can be empirically determined. And in this case, we'll end up using a little bit of both. We'll use our empirical data we learned from phase 2, and then we'll also end up estimating the Z values of F and A based on literature or several other models and information that we can find. So then step 3 will be determine D values or movement rates of these. And there are 2 different types of what I call movement rates that can occur here. One is advection, where a chemical being transferred between 2 phases undergoes no conversion between that chemical structure. It remains the same, or if it's genistein in one phase, it will remain genistein in another. The other type of movement that can occur is reaction rates, where the chemical is converted through some process going between the phases. If it is uptaken by a fish, microbial action in the gut can take it from genistein and potentially turn into glucoside, or vice versa. So basically you have to map out what are all the potential areas, or all the potential movement that can occur from this chemical in the water. So obviously we have water coming into the system, since this is a aquaculture system. You have outflow of the water occurring. You have pellets being introduced, which will carry the chemical into the system. So then the question is it being leached out into the water? What is all being uptaken by the fish via consumption or even potentially respiration? And then once it's in the fish, how much is being bound up within the fish or being lost through excretion, respiration, growth dilution, or egestion? And then along with that, we also have to include the potential for any air-water reaction, the volatilization or reabsorption back into the water that occurs. So our final model is a nice big mess of arrows and stuff. But basically, from this, we can end up calculating the overall fugacity of the system by knowing the overall amount of chemical being introduced to the system and dividing that by all of these transport rates throughout the system. And then once we know the fugacity of this model, we can use that initial equation to determine the amount of concentration that will occur within each of our compartments using the ZF formula. And then by using volumes, we can also determine just the straight amount of chemical that can be found there. So while we have a model, we know that all models are not they're not perfect. They cannot 100% give you accurate results. So what we like to do then is perform an independent feeding trial in order to determine the accuracy of this model that we are developing. So in order to do this, we will be using a feeding trial looking at 4 different diets. There will be those phase 2 diets again that come back, and we'll also use a reference diet that does not have any genistein or soybean meal included in it. We'll be utilizing swim up rainbow trout, and they'll be fed for a duration of 112 days. And I selected this time period because this is when these fish are experiencing their sexual differentiation period, and genistein should most likely have the greatest effect at this point upon the reproductive development of these fish. These fish will be fed 3 times per day and they'll be fed to satiation. And this will be performed in a flow-through system consisting of 4 replicate aquaria per diet, and they'll be stocked with 150 fry per aquarium. So there are numerous metrics we want to look at here. On a daily basis, we are going to determine the amount of consumption of feed based off of visual observations in order to help us determine feeding rations, so we're not giving them too much with a great amount of excess feed in the system. And along with that, we can use those and also to determine food conversion ratios and protein efficiency ratios, which basically gives us a grasp of how well can these fish utilize these diets. So then we'll also, on a bi-weekly basis, we will take total tank weights, or to find out the total biomass, and also determine volume by a displacement method. This will be important for determining our feed rations and also the overall weight gains to see how well can these fish grow on these diets. And along with that, this will also help to tell us how much chemical is being incorporated into the system and what are the volumes of the fish within there, which becomes necessary for our calculations later on. Water quality will be measured 2 different ways. We'll have daily measurements of just your basic water quality parameters in order to determine we have adequate culture conditions. Along with that, we'll take bi-weekly samples in order to determine the levels of genistein within that water for our modeling purposes. So for some physiological parameters, we will take out 10 fish on a decelerated sampling period, which basically means at the start of this experiment, we'll take 10 fish out 1 time a week, and then because they'll be growing much faster with a greater chance of incorporating those chemicals, as our growth slows, we will then extend out those periods to every 2 weeks until the end of the experiment. We'll look at whole body genistein and vitellogenin instead of plasma due to the small size of these fish. And at the completion of the 112-day feeding trial, these fish will then be moved to a larger system and held for an additional 1 to 2 years to allow for the gonads to reach full maturation or full maturity. And then from that, these fish will be necropsied, and we'll take blood samples to determine any sex steroids for any secondary characteristics that may have occurred from the inclusion of genistein, and also histology slides to look for those intersex species, or for those intersex characteristics. And these will primarily be focused towards the male fish. So just some basic stats for our growth and some of our physiological metrics. We will use a one-way analysis of variance again with a LSD post-hoc. For the Fugazi model, we will analyze in order to determine its overall accuracy, we'll use 2 different methods. The first will be paired t-test, looking at the estimated values against our actual values. And the second will be a decomposed mean square that looks at the differences in slope of between our estimated and actual values, along with the slope differing from unity and the random variation within the system. So I'd just like to thank SDSU for taking me on and letting me perform this research. Along with that, the USGS for hiring me and allowing me to run these experiments at their place, and for Prairie Aquatech for the help with the diet development and information there. With that, I would take any questions you may have.
What about solubility is usually dependent on temperature?
Yeah.
What about water temperature for warm water fish propagation versus like trout in cool water?
Yeah.
Do you have any plans to address that issue?
Not currently. The main reason we're doing cold water fish species is due to the fact that in the literature, a lot more research has been done on the rainbow trout and the salmon species, and the fact that they are relatively easy to get and they are quite a good guinea pig to use. So at this point, no, not yet. But ideally, if this model does work, it would be hopefully beneficial then to look at it in that way and use it with some warm water species as well.
And how many parts per million of genistein is needed to start causing some of these sex change kind of things?
Oh boy. Oh, that's right. So the ones that I use, the 2 milligrams per gram and 4 milligrams per gram, that's what causes intersexes in a channel catfish
So
species.
2 parts per million?
It was, yes, 2 parts per million. It was in both of them, 2 and 4.
Why do we take this feed conversion and protein conversion efficiency daily, is there any, is it very stringent needed, that daily measurement?
Say that one more time.
This measurement of feed conversion ratio, protein efficiency ratio on daily basis.
On a daily basis?
Should we need to do that, or we can prolong the day duration, maybe weekly or?
Well, yes and no. We'll still need to take it daily due to the fact we need to know how much chemical is going to be introduced into the system. So the data is already going to be there in order for that. So we might as well just estimate it for each day since we'll be taking it anyway. I should repeat that. Okay.
We do have a message coming in from West River. Might be able to take another quick one before it actually arrives. Type faster, Kurt.
Come on, Kurt.
Hi, sorry, here I'll just say it. So one of the things that you talked about at the beginning of your presentation was that there's concern of the water and waste products of aquaculture getting into the environment. And I wonder how you might address how fish in the environment might be uptaking these estradenoids, or I'm not saying that properly, but do you understand what I'm asking?
To a degree. So basically you're wondering, well, I'm not sure if there's been much concern. It's just that nobody's actually looked at this, so we really don't know if enough is even getting out into the environment at relevant levels. So this is just one way of looking at, is there even a possibility that we'll see that getting out there, and at what level? And if we know that, we could possibly determine off of experiments. If we know the amount of level that is potentially being introduced into the environment, we could then say at this level we could do some tests to see if that's a relevant level that will create these biological effects. But currently we just don't know, so we're just trying to determine actually how much could potentially leave our system and get into the environment.
Thank you.
All right.
So if the levels are high enough, what methods are there to probably inactivate?
That's part of that phase one. The processing techniques you can use, there's a potential that you can adjust them to either decrease it and get more of the isoflavone into the secondary products possibly, but we currently don't know. Some of those food processes that I was mentioning, they actually saw they could reduce like the isoflavones in the final product by up to 74% just by, but most of that goes into the wastewater or into like the secondary products. So instead of getting into the tofu that we're going to eat, it gets into some of this extra items that aren't used or are just flushed down the drain. Which also brings up the question for flushing them down the drain, where are they going then? So.
All right, everyone, let's thank both our speakers one more time. So next week we'll have one presentation, and then we will also have the University IACUC representative visiting with us to talk a little bit about policies and procedures. Thanks, everyone.